Welcome back, ladies and gentlemen. So last time we were talking about prokaryotic gene regulation, where they pretty much do all of their regulation at the level of transcription, right? Either they produce those messenger RNA molecules or they don't. When we get into multicellular organisms, we've got a whole lot more options as to where we can regulate that gene expression, and it's necessary for cellular specialization as well as just efficiency processes. So we are able to really drill in to that gene expression and regulate it at a whole host of levels, a whole host of steps across that process of transcription and translation. So when we look at your cells in your body, for example, all of your cells in you are genetically identical. I pull out a cell from your skin versus a cell from your heart versus a cell from your brain, and your genome is more or less going to be identical, right? Your sequence of A's, T's, C's, and G's is the same. Each of those cells contains 46 chromosomes, and the information within those 46 chromosomes is the same. So how do I differentiate? How does your cell recognize that I am supposed to be a brain cell, I am supposed to be a skin cell, I am supposed to be a muscle cell? That difference between cell types is a result of differential gene expression, where I'm expressing different genes by different cells, but every cell in your body has the potential, at least at some point within its life cycle, to have the information from your entire genome. We get errors in that gene expression, and that's where we get diseases like cancer. So gene regulation is incredibly important, and it's also incredibly important that as multicellular organisms, we have a lot of places to do this. So I can regulate it directly at the structure of the chromosome itself. Is it loose and spread out as chromatin, or is it condensed in terms of access to that information? I can regulate it at the transcription level. I can regulate it at the RNA processing level. So we talked a little bit about alternative splicing with introns and exons. So I can regulate it there. I can regulate it at the ability for it to get exported from the nucleus. I can regulate it around translation, right? Breaking down that messenger RNA or just regulating how much of it works. I can regulate it at the protein processing level. I can regulate it where that active protein actually gets put in that cell or gets exported from that cell. So anywhere along this pathway, from access to the information to the ultimate actual cellular function of the finished protein product, we have places to regulate the gene expression. So we're going to start today in this video just talking about the regulation of the chromatin structure itself. So remember that your genes, when we looked at cell division, we talked about DNA in your cells being loosely wound around those proteins called histones, right? And those histones can sort of be sticky, or sometimes I use the analogy that they can be magnetized or demagnetized to allow us to condense all of that chromatin when it's time to move it during cell division into that super hypercoiled tight chromosome structure that we're used to seeing, those big X's, right? So when we talk about gene expression, well, we spread out that chromatin during interphase because I want access to the DNA. I need to know how my cell needs to function, and it needs access to those instructions. When I go through cell division, I want to condense everything down because all I'm trying to do is move it and not break it. But when we look at your cells during interphase, when they're doing their normal function, not 100% of that DNA is actually spread out in chromatin structure. So what we'll do is we'll take certain components of your chromosomes that aren't necessary for that cell's function and condense it, right? While the genes that I do need for that cell to do its job are left loose and accessible. So if I am a cardiac muscle cell, the genes necessary for me to do my job as a cardiac muscle cell are the ones that are loosely wound in sort of that traditional chromatin structure, and now I have access to it. But when I look at that cardiac muscle cell, I'm going to condense the information and turn off access to the information on how to be a neuron because that's not my job. I'm going to turn off the information on how to be a skin cell because that's not my job. I go to that neuron, however. Now that neural information on how to be a neuron is loose and accessible, and the stuff to be a cardiac muscle cell is highly condensed and not available. So we look at chemical modifications to these histone proteins, as well as to the DNA nucleotides themselves that can influence both the chromatin structure and, as a result, the gene expression. So one of the things we can do is utilization of acetyl groups, so CH3CO. These acetyl groups can attach to those histones and specifically attach to positively charged lysines, right? Lysine is an amino acid that exist in these histone tails. So here are those histone balls, right? They also have these little tails that come off of them, and then the DNA double helix is wound around it. If I attach those acetyl groups to this, right, this can help to loosen that chromatin. So basically, if we think about these guys as magnets, adding those acetyl groups, to those lysozyme tails demagnetize the histones and they're all spread out. Okay. The converse attachment to that is to add methyl groups instead via a process called methylation. Adding those methyl groups will do the opposite. So they will actually condense the chromatin. So I have this interplay between acetyl groups and methyl groups that their presence or absence has a dictation as to whether or not we have this condensed version of that chromatin blocking access to it, or I have this loose version of chromatin where I now have access to that gene. So presence of acetyl groups loosens the chromatin, promotes the initiation of transcription of that gene. Presence of methyl groups condenses it, prevents the transcription. I can also use phosphate groups via phosphorylation next to a methylated amino acid and then loosen the process again. So acetyl groups loosen, phosphates next to a methyl group loosen, methyl groups themselves condense, lack of acetyl groups condenses. Okay? And so we talk about this idea of this histone code hypothesis, which sort of proposes that the combination of modifications made to these histone tails helps to ultimately determine chromatin configuration and as a result influences the ability for transcription to occur or not, right? So loose chromatin I should have access to, that should allow transcription to occur to create the messenger RNA for that set of instructions within that piece of loosely wound DNA. Condense it all, I shouldn't have transcription happening really at all because I don't have easy access to the DNA molecule. We can also add methyl groups directly to DNA itself, and this will also reduce transcription. The interesting thing is when we look at methylation of histones, this is a lot more fluid, right? It's a lot more malleable. It can happen. We can relatively easily take them off. We can pretty easily make modifications to these histone tails. On the opposite side, if we methylate the DNA directly, this often is much more long-term. So when we're talking about more environmental influences, right, temporary environmental influences, we may be doing more histone modification. But if I have environmental influences that are causing changes directly to the DNA, methylating the actual DNA itself, this tends to be much more long-term, and we see this much more associated with genes that are resulting in cellular differentiation. It's not to say that the DNA methylation never happens, can never come off, but this is much more associated with permanent gene turnoff. So when we talk about genomic imprinting, this is methylation that's going to regulate expression of either maternal or paternal genes. So when we go through and look at genetics and we talk about your typical Mendelian genetics, right? I got the dominant trait from my mother on that chromosome, the recessive from my father, so I should express the dominant trait because I'm heterozygous for it, right? Interesting thing is, there are certain situations where it doesn't matter the combination of genes that you get. What matters is which parent gave it to you. And that's this idea of genomic imprinting that we see a lot more with the methylation directly of the DNA itself. So even though mom gave you dominant, dad gave you recessive, the way that that particular gene's expression works is that you always express the paternal allele, and so dad gave you recessive, and so you are recessive for that particular trait, regardless of what your mother gave you, because we methylate mom's gene and shut it off. Hopefully that makes sense. Point here is that the expression of your genetics is way more complicated than just traditional Mendelian genetics for most of the traits that we see. So the chromatin modifications don't actually alter the DNA sequence. All they do is alter in that particular cell, in that particular individual, whether or not it's being expressed. So that does mean that we can still pass on that genetic information to future generations, to future cells. And so the expression of genes is way more fluid than we used to think. We used to think that your DNA was your DNA was your DNA, and you were going to express it, period. We now understand that within your own body, this methylation, this acetylation, can influence your expression of your set of DNA. And often those methyl groups, those acetyl groups, are influenced by your environment, what you eat, what sort of pollutants might be around. The cool thing is that that also means that changing your diet or changing your lifestyle, getting away from pollution, may also mean we can alter the way your genes are being expressed from one point in your life to another. And that's this idea of epigenetic inheritance, is that we see the inheritance patterns not only playing out simply because of the sequence of A's, T's, C's, and G's that you have, but then we add on this extra layer of inheritance on top of that and gene expression on top of that in the sense of this epigenetic inheritance. And not only can we see that epigenetic inheritance within a single organism, some of that epigenetic inheritance also seems to get translated from one generation to the next. So what your mother ate while she was pregnant with you may have an effect on your children down the line. There's a really neat article that I have linked on Schoology for you about trauma from Holocaust victims and how we think there might be epigenetic connections to those experiences, the starvation, the depression, the you name it, all of their experiences going through things like being in a concentration camp, having potential implications to the mental health of not only themselves, but their children and their grandchildren and possibly their great grandchildren. So, this idea of epigenetic inheritance is really pretty cool because not only does it influence you, but it influences your future generations. But on the flip side to that, we have some control over changing certain environmental factors to sort of reverse that inheritance as well, which is.
You ever notice that if you get to know two identical twins, they might look alike, but they're always subtly different? Yeah, whatever. As they get older, those differences can get more pronounced. Two people start out the same, but their appearance and their health can diverge. For instance, you have more gray hair.
No, I don't. Identical twins have the same DNA, the exact same genes.
Yeah.
And don't our genes make us who we are?
Well, they do, yes, but they're not the whole story. Some researchers have discovered a new bit of biology that can work with our genes or against them.
Yeah. You're heavier and I'm better looking.
Yeah, whatever. Imagine coming into the world with a person so like yourself that for a time you don't understand mirrors.
As a child, when I looked in the mirror, I'd say, "That's my sister." And my mother would say, "No, that's your reflection."
And even if you resist this cookie-cutter existence, cultivate individual styles and abilities like cutting your hair differently or running faster. Uncanny similarities bond you together. Facial expressions, body language, the way you laugh, or dress for an interview perhaps, when you hadn't a clue what your sister was going to wear. The synchrony in your lives constantly confronts you.
When I see my sister, I see myself. If she looks good, I think, I look pretty today. But if she's not wearing makeup, I say, my God, I look horrible.
It's hardly surprising, because you both come from the same egg. You have precisely the same genes. And you're literally clones, better known as identical twins. But now, imagine this. One day, your twin, your clone, is diagnosed with cancer.
Por favor.
If you're the other twin, what can you do except wait for the symptoms?
I have been told that I am a high risk for cancer. Damocles' sword hangs over me.
And yet, it's not uncommon for a twin like Anna Marie to get a dread disease, while the other, like Clotilde, doesn't. But how can two people so alike be so unalike? Well, these mice may hold a clue. Their DNA is as identical as Anna Marie and Clotilde's, despite the differences in their color and size. The human who studies them is Duke University's Randy Girdle. So Randy, I see here you have skinny mice and fat mice. What have you done in this lab?
Well, these animals are actually genetically identical.
The fat ones and the skinny ones?
That's correct.
Because these are huge.
They're huge.
Can we weigh them to find out?
So if you take, this is...
Looks like they can barely walk.
They can't walk too much. They're not going to be running very far. So that's about 63 grams.
63 grams.
Let's look at the other one.
So it's half the weight.
Right.
This gets even more mysterious when you realize that these identical mice both have a particular gene called agouti. But in the yellow mouse, it stays on all the time, causing obesity. Just look at this. So what accounts for the thin mouse? Exercise? Atkins? No. A tiny chemical tag of carbon and hydrogen, called a methyl group, has affixed to the agouti gene, shutting it down. Living creatures possess millions of tags like these. Some, like methyl groups, attach to genes directly, inhibiting their function. Other types grab the proteins, called histones, around which genes coil, and tighten or loosen them to control gene expression. Distinct methylation and histone patterns exist in every cell, constituting a sort of second genome, the epigenome.
Epigenetics literally translates into just meaning above the genome. So if you would think, for example, of the genome as being like a computer, the hardware of a computer, the epigenome would be like the software that tells the computer when to work, and how much.
In fact, it's the epigenome that tells our cells what sort of cells they should be. Skin, hair, heart. You see, all these cells have the same genes, but their epigenomes silence the unneeded ones to make cells different from one another. Epigenetic instructions pass on as cells divide, but they're not necessarily permanent. Researchers think they can change, especially during critical periods like puberty or pregnancy. Schertl's mice reveal how the epigenome can be altered. To produce thin brown mice instead of fat yellow ones, he feeds pregnant mothers a diet rich in methyl groups to form the tags that can turn genes off.
And I think you can see that we dramatically shifted the coat color, and we get many, many more brown animals.
And that matters because your coat color is a tracer. It's an indicator.
That's correct.
Of the fact that you have turned off that gene.
That's right.
This epigenetic fix was also inherited by the next generation of mice, regardless of what their mothers ate. And when an environmental toxin was added to the diet instead of nutrients, more yellow babies were born, doomed to grow fat and sick like their mothers. It seems to me this has profound implications for our health.
It does for human health. If there are genes like this in humans, basically what you eat can affect your future generations. So you're not only what you eat, potentially what your mother ate, and possibly even what your grandparents ate.
So how do you go to humans to do this experiment when you have these mice and they're genetically identical on purpose?
That's right.
So who is your perfect lab human?
Well, then we look for identical humans, which are identical twins.
Twins. And that brings us to the reason why we're showing you Spanish twins. In 2005, they participated in a groundbreaking study in Madrid. Its aim? To show just how identical epigenetically they are or aren't.
One of the questions of twins is that if my twin has this disease, I will have the same disease. And genetics tell us that there is a high risk of developing the same disease, but it's not really sure they're going to have it, because our genes are just part of the story. Something has to regulate these genes, and part of the explanation is epigenetics.
Esteller wanted to see if the twins' epigenomes might account for their differences. To find out, he and his team collected cells from 40 pairs of identical twins, aged 3 to 74. Then began the laborious process of dissolving the cells until all that was left were wispy strands of DNA, the master molecule that contains our genes. Next, researchers amplified fragments of the DNA until the genes themselves became detectable. Those that had been turned off epigenetically appear as dark pink bands on the gel. Now, notice what happens when the genes from a pair of twins are cut out and overlapped. The results are far from subtle. especially when you compare the epigenomes of two sets of twins that differ in age. Here on the left is the overlapped DNA of 6-year-old Javier and Carlos. The yellow indicates where their gene expression is identical. On the right is the DNA of 66-year-old Anna Marie and Clotilde. In contrast to the younger twins, hardly any yellow shines through. Their epigenomes have changed dramatically. The study suggests that as twins age, epigenetic differences accumulate, especially when their lifestyles differ.
One of the main findings of research is that epigenomes can change in function of what we eat, of what we smoke, of what we drink. And this is one of the key differences between epigenetics and genetics.
As the chemical tags that control our genes change, cells can become abnormal, triggering diseases like cancer. Take a disorder like MDS, cancer of the blood and bone marrow. It's not a diagnosis you'd ever want to hear.
When I went in, then he started patting my hand and he was going, your blood work does not look very good at all, and that I had MDS, leukemia, and that there was not a cure for it, and basically I had 6 months to live.
Was epigenetics the reason? Could the silencing of critical genes turn normal cells into cancerous ones? It's scary to think that a few misplaced tags can kill you. But it's also good news, because we've traditionally viewed cancer as a disease stemming solely from broken genes. And it's a lot harder to fix damaged genes than to rearrange epigenetic tags. In fact, we already have a few drugs that will work. Recently, Sandra Shelby and Roy Cantwell participated in one of the first clinical trials using epigenetic therapy.
The idea of epigenetic therapy is to stay away from killing the cell. Rather, what we are trying to do is diplomacy, trying to change the instructions of the cancer cells, reminding the cell, hey, you're a human cell, you shouldn't be behaving this way. And we try to do that by reactivating genes.
The results have been incredible, and I didn't have really any horrible side effects.
I am in remission, and going in the plus direction is a whole lot better than the minus direction.
In fact, half the patients in the trial are now in remission. But while it may be easier to fix our epigenome than our genome, messing it up is easier too.
We've got to get people thinking more about what they do. They have a responsibility for their epigenome. Their genome they inherit, but their epigenome they potentially can alter, and particularly that of their children. And that brings in responsibility, but it also brings in hope. You're not necessarily stuck with this. You can alter this.